Yeah, hi everyone, welcome. This is going to be the next session of the Omics workshop, so session number 7, Intro to Omics. Before we get started, just a really quick announcement. We officially have some Omics Club merch. So we designed a t-shirt. Gloria Stoyanova designed it. Kevin helped to put it together for the sale. It's a charity that's supporting, it's a charity fundraiser that's supporting, which charity is it, Kevin? Bronx Works. So, right, okay, that's the local Bronx charity that supports like low-income families and development in the Bronx. So it's yeah, it's pretty exciting. It's a cool design. So yeah, look out for your email for a link to that. So with that, I will go ahead and hand it over to Isaac.
Can you all hear me? All right, it is my pleasure to introduce Dr. Simone Sidoli. He's an assistant professor in the Department of Biochemistry. He's worked in the fields of proteomics, mass spectrometry, and chromatin biology for about 15 years. He trained in the University of Parma in Italy before doing a PhD at the University of Southern Denmark and a postdoc at the University of Pennsylvania. His group studies the changes in conformation and architecture of chromatin during aging using cohorts of centenarians here at Einstein, as well as developing 3D cell culture model systems. Dr. Sidoli also runs the Einstein Proteomics Core, which serves nearly 200 different users per year. It's also my pleasure for the second half of today's workshop to introduce Juan Sepulveda to lead today's vignette. Juan completed his bachelor's in biological sciences at Cornell University, where he studied the coevolution of vertebrate animals alongside their microbiomes, with a particular interest in the impacts of temperature and urbanization. He pursued his interests in computational biology during his rotations at the labs of Dr. Yunhao Wu, Matthew Gamble, and Roger Chang. Juan joined the Chang Lab in the Systems and Computational Biology Department, where he studies the role of metabolism and neurodegeneration, and particularly glaucoma. Today, he will introduce you to the common data processing strategies used in proteomic studies and provide the foundations for the interpretation of such data. So without further ado, Dr. Sidoli.
Thank you, Isaac. Thank you, everybody, for being here. And I'm going to be very brief with my part because I want to give enough stage to Juan, who is, the real reason why you guys are here to really process a proteomic data set. Well, first of all, let me thank the Omics Club for the invitation and congratulate them because I think they just created the most popular course that Einstein ever had. The other courses, they don't give sandwiches, so it's a bit unfair. But yeah, what I would like to do with you before we talk about the real data, I just would like to spend a few minutes giving you an overview about what proteomics is, and you probably already know. So I hope I can spend the next few minutes giving you a slightly different perspective from what you are familiar with. You think about proteomics. Let me see it from here because that one is a bit far for me. Yeah, you think about proteomics as studying all proteins, right? In a biological system, which can be a cell, a tissue, a complex organism, or other kind of samples that we analyze all kind of strange samples. And obviously, you know that proteins are not RNAs, but you are used to think about proteomics like the study of proteins that is basically the equivalent of studying RNAs, or just a different kind of biomolecule. Now, I would like you guys to think about the proteomics as something that is slightly different than studying RNAs or other type of transcripts. And I wrote it here, summarizing in this slide. Think about the difference between differentiating what a cell has versus what a cell produces. Right? So when you do transcriptomics or you study the structure of the DNA with ATAC-seq or whatever, you think about what that cell is doing in that precise moment, right? When you do proteomics, a little bit different, right? And I like to use this example to put it in perspective, right? So one of the most abundant protein in our blood is serum albumin. Like we all know serum albumin. Blood does not produce serum albumin. It's produced by the liver. When you study a cancer tissue, like a lot of proteins that you see in that cancer tissue come from immune cells. So when we do proteomics, especially in a complex organism, what we're looking at the protein content in the sample that we analyze, those proteins in that sample are not necessarily coming from those cells. Like many of them, of course, they do, but not all of them. So when you take proteomic snapshots of whatever sample you're studying, you are actually looking at what is happening in that context, regardless of who produced these proteins. And that, if you think about it, is already a different perspective than thinking about RNAs, right? Because usually RNA, they don't travel as long. And usually when you study transcriptomics, you're looking at what that cell specifically is doing in that moment. Now, compared to sequencing, proteomics is definitely less sensitive, is definitely less reproducible. You guys probably in the previous sessions, you have been working with the p-values of 10 to the minus 100. They don't exist in proteomics. Like our accuracy in measuring protein abundance is not nearly this good, right? But we actually have other kind of advantages. And I'm going to tell you in a couple of slides. First of all, I know you probably have seen this before, like many of you attended the biochemistry courses, like because you guys are young students. I've been showing this for a while. A typical proteomic workflow, just for everybody to be on the same page, is usually based on mass spectrometry. So we extract proteins from nearly anything. We have projects in the lab when we analyze proteins from breath, from earwax, not the majority, luckily. But what we do from these proteins, once we extract them, we digest them into shorter peptides, and then we run them into the mass spectrometer, which is usually coupled online with liquid chromatography. This is one way of producing proteomics data, is by far the most popular way. It's not the only way. So I just want to spend just 1 second, making you all aware that there are other methods to produce a proteomic data set. Some of them are based on microarrays, for example, like aptamers. Some of them are based on antibodies that are tagged with DNA, and therefore they can multiplex, to study many, many proteins in once. And some of them are based on, again, antibodies, but they recognize very short pieces of proteins, and once they are combined together, they build the protein sequence for you. And the top two are commercially available. They're actually not as rarely used as you might think. The one below is an example of a method that is still a little bit in development. But bottom line, you can produce like long list of protein identification and abundances also with techniques that are not mass spectrometry. The problem with most of these methods is that they tend to be more biased. So because obviously when you have a microarray, you can only quantify proteins that are for which you have baits in your chip, or antibodies, like same concept. Mass spectrometry instead looks at different perspectives, and it tries to be more unbiased. And about that, I actually had some fun putting together this slide. If you think about an experiment that you can do with mass spectrometry to look at proteins, you can actually look at so many quantitative dimensions, like many of them in the same experiment, that it really, the goal of us being here today is really for you to think about how flexible it can be your project design, so that it gives you a perspective on your biological system that you couldn't observe before. Now, obviously, we don't have time to go through all of this, but just to give you an example. Now, obviously, today with Juan, you guys are going to talk about protein abundance, right? Because the table that you have has a certain intensity value that corresponds to the abundance of the protein. You guys are going to calculate fold changes, how much this protein changes. You guys are going to calculate p-values, how reproducible these changes are. But then you see there are all of these other possibilities, like things that you can do. You can look at protein co-regulation. You can look at translocation. For example, if you fractionate organelles and you do proteomics of these different fractions, you can look at protein synthesis rate. So if you put a metabolic label into your system, and then you actually quantify how rapidly that label is being incorporated in your protein, you are no longer looking at how abundant the protein is, you are looking at how rapidly it's being synthesized. So that's another perspective. We're not going to talk about this, like we're going to focus on the first three today. I just wanted to shed light on the fact that we can be very, very flexible when we design an experiment in proteomics, in particularly when we are using mass spectrometry. So speaking of this first 3 dimension, there is something else that is maybe not as obvious to scientists that are used to work with sequencing data. We are used to work with RNA. And one of these concepts is that proteins have an extremely wide dynamic range. If you look at proteins in blood, serum albumin can be as abundant as 12 orders of magnitude more than some very, very low abundant proteins that are present, in our blood. 12 orders of magnitude. I don't know if it's, easy to imagine this magnitude. You think about it in space is more or less as the size of a bee and the distance between Earth and the moon, or even more. In time is the distance between my age and the age of the universe, right? So there's no technology that can look at things that are 12 orders of magnitude apart. And that includes mass spectrometry, unfortunately. So part of the problem that we have when we analyze proteomes is that we don't see all proteins because they span such a wide dynamic range. We have methods to fractionate the sample to try to improve our sensitivity. But when you work with a proteomic data set, you always keep in mind that you're always missing something. There's always something missing. And even when we work with single cell proteomics, that I'm sure you've heard of it, yeah, we can look at proteins from very, very tiny amounts of material. The problem, again, is not the sensitivity of the instrument, it's the dynamic range of the instrument. The very abundant signals, they suppress the ones that are very low abundant. So keep that in mind. But in a way, it's also a very interesting perspective to look at. If you actually have a look at this graph that I put here on the right for you, these are, this is just a proteomic data set like another. These are protein ranked by abundance. And they are divided in quartiles, like the different colors that represent basically 25% of the protein hindrance. And then you can see that, for example, there are some samples, like some tissues, where 1 or 2 proteins occupy 25% of the protein mass, or sometimes 50% of the protein mass. If you take a muscle cell and you want to do proteomics, let's say you do transcriptomics on that, you can probably easily see 10, 15,000 RNAs. If you do proteomics of that, 95% of that proteomic data is myosin, right? So that's, the more a cell is specialized, the more a few proteins define almost the total mass of the cell occupancy. So that is something else to keep in mind. And then obviously, you guys today are going to discuss about fold changes and p-values, which are the other 2 dimensions that I quickly discussed. Now, this one is intuitive. I'm not going to spend that much time on, but I'm going to promise you that by the end of the lecture, you guys will be able to produce those volcanoprods that you can use to organize your tables, and you will be able to tell from an analysis which ones are the proteins that are probably the most interesting for me. The only thing I want to say about this slide, just a little piece of advice, always treat p-values as detectives and not judges, right? Because that's a common mistake that we do when we think about p-value threshold. Like, so we think that everything that is lower than 0.05 is good, and everything that is 0.06 and higher should be trashed. P-values are calculations of a probability, right? If I tell you the story of where the t-test comes from, that is something that is very useful, especially to sort your tables. Is not something that should be used to judge the quality of the data for you. And the last thing I wanted to mention, since we are stuck on these 3 dimensions, we're not going to go beyond it because of the type of table that you guys are going to handle. Consider that these 3 dimensions can also be looked at together. So never forget, and this is probably true for RNA data as well, never forget that when we look at fold changes, we tend to disregard how abundant a certain signal is. And then, as I told you before, how much protein you have in a sample actually can make a heck of a difference in terms of what is the phenotype of that cell, right? So think about a protein that is changing twofold, right, in a muscle tissue. If that is myosin, you're talking about millions of copies of proteins that are being produced, more or less. That actually means something, right, rather than something that is present in a very, very low abundance. What is low abundance is not less important, but it's important to include this in giving you this perspective. And the reason because I included this graph, actually this graph is not a volcano plot. This graph actually has on the two axes, on the x-axis, the fold change, on the y-axis, the protein abundance. And then, the color is the p-value. But what is interesting, at least in my perspective, in a graph like this is how, for example, we represent data when we look for biomarkers. So if you have a new disease and you want to find whether there are proteins that are specific for that disease, and you want to actually develop an assay to be able to quantify this protein, for example, by ELISA, like very rapidly once you have lots of patients coming into the clinic, it's important to consider also how much of that protein you have in the biospecimen that you purify, rather than just how enriched it is. So you guys, you will consider all three dimensions as you analyze your data today. And actually, that's it. Again, I didn't want to take too much of your time. I want you guys to be able to have enough time to play with your tables. Just some final notes, and I don't know if there's any question. If not, I'm going to let Juan take over. Any question? Yeah. I was working on almost like yeah, actually, that's a very good question. And I think we partially address with the way that we're going to process the data today. So the issue with filtering, is always that there is no weight on which features seems to be the most enriched. Right? So you're right. Like if we filter for like two folds and p-value 0.05, you end up with 500 proteins. Like what do I do with that? Now within those 500 proteins, some of them are vastly more regulated than others. Then all of these 500 proteins contain information. I don't know if we're going to discuss it today, but obviously we can group them into functions. We can group them into pathways. We can find like some common features that they have. But we should never forget that these 500 proteins, they are not all equally regulated in the same way, right? So there will be some that are definitely more enriched in whatever comparison you have compared to others. And Juan will address this issue as well. I don't know if we're going to talk about biological interpretation a little bit. Yeah, so hopefully that will answer your question. If not, we can keep talking. Yes. Yeah, that's a good question too, right? So, and that's actually the reason of why proteomic quantification is not as accurate as transcriptomic quantification, right? So when you analyze transcripts, I'm not an expert, but you have all of these sequencing reads, like you put them on top of each other, you count them until you have like an area, and that tells you the intensity of your signal. When we do proteomics, what we do, we have profiles in our chromatograms, right? And then when the signal comes into the instrument, you get a signal intensity. That value corresponds to what we estimate to be the peptide abundance that then is converted into protein abundance. But because those chromatographic profiles, they are supposedly have a Gaussian distribution, like you need to be able to tell where that signal begins and ends to tell, okay, this is the intensity of my peptide. These profiles, they never look as good as you might think. And so it's much harder to tell what are the boundaries of my peak, what is the real intensity of my signal compared to counting reads. So at the end, you will see that the variability in a proteomic dataset is always higher than a transcriptomic dataset, mostly because of the nature of how we quantify. That's it? Perfect. Okay, so now you guys are up for a ride.
All right, guys. So we're gonna get set up here for the next part of the session. So we'll probably take like 5 minutes to do that. So I guess if people have to run to the restroom or anything, feel free to do that. So yeah, we'll circle back in 5 minutes.
Actually, I also have a suggestion. During the break, I strongly encourage you all to go into the Omics Club GitHub, download all the files, because this will be an interactive session. You will be able to run the code. So I do encourage you to download the code and the files before we start. Yes.
Yeah, we'll do a walkthrough of that once we get started. Testing, testing, testing.
I will also make the announcement that I have to leave at 1:25. Oh, really? Because that's when my class begins. Oh, really? Yeah, so we have. So yeah.
Testing, testing. Can you guys hear back there? In the back, you can hear? Okay, nice. Yeah, so I guess we'll go ahead and get started. Yeah, our next speaker is Juan. So we already read the intro for that. So there you go. Please take it away, Juan.
Hi, everyone. As you all know, I'm Juan, and I'll be leading today's vignette. So I'll walk you through every single step to make this as interactive as possible. But before I start, I do want to survey you all to get a sense of where we all stand. So can I get a raise of hands for people that have coded in Python before? Okay. Can I get a raise of hand of people that have coded at all? Okay. And can I get a raise of hands of people that have used Excel to do just to do formula and stuff like that, that feel proficient with it? Okay, that's wonderful. Okay, so everyone should be able to follow. So first we'll start with going to the Omics Club GitHub. I hope everyone has a link to it. So we start with the Omics workshop, we go to the Proteomics, and these are all the files I would like you all to download. Can I also get a raise of hands for who has all these files in their computer right now? Okay. Yeah, and this is how you download them. Basically, you go. Actually, how do you download it? Help. How do you download things from GitHub? Top right. Oh, that's top left. Sorry. And then should I? Okay. Yeah. So I guess you can download it individually, and yeah, so please do that. And I'll show you what are in these files. Yep, okay. So I have downloaded most of these files in my computer, and this is how they should look like, probably without this, without this, and without this. So I'm gonna walk you through what's in each of these files. So this one is for people that haven't coded before and kind of just want to follow along with their Excel knowledge. What's inside this file as it opens. Is this sheet, actually workbook with multiple sheets. And we're going to basically walk through with the code what is being done in the Excel sheet. So essentially everything that I'm doing with Python is being done in Excel. So you should be able to follow along. What we have in this file is the raw data, which are the accession code. So at this point, we're assuming that you've done your proteomics study, you've sent your samples to the proteomics core, and you've gotten back a file that has the accession number for every protein that has been found in your sample. Potentially some metadata, description of the proteins, the gene symbol of the proteins, and the organism from which that protein comes from. Additionally, after you submit your file, you might end up getting these abundance reads for the different conditions you might be trying to measure. In this case, as you can see here, this data comes from this experiment where 2 conditions were done in 4 replicates, and then 2 of those replicates were acquired in a single experiment. So that's why we have this split of 1.1, 1.2, 2.1, 2.2, same thing for HSV. The control, as you might assume, are healthy cells, and HSV are cells that are infected by this virus, HSV herpesvirus. Okay, so far so good. What we're going to do through this vignette is take this raw data into things that you can actually interpret and give you the tools on how you can interpret beyond the first 2 dimensions that Simone talked about, more on the interpretation. But as we keep going and as I keep showing you this workbook, we have different questions we want to address during this vignette. So we want to look at how the raw data looks, how after we transform the data, how that also looks. And then one of the more important sheets in this workbook is the data processing. This is what my code is actually going to be doing. I essentially translated Excel code onto Python code. And the way that I've structured the Python workbook is meant to mirror what is going on here. So we start with our raw data, basically the same things from here. And then we're going to start doing certain types of transformations. I will explain why we do these transformations and the result of those transformations. But in the Excel sheet, you can see how those transformations are made for each column. So we're going to do something called the log2 transform. Then we're going to do normalization. We're going to impute. And we're going to generate a volcano plot. And for the volcano plot, we require these different parameters. And we'll get to how they're done. We are also going to generate figures. They should reflect the figures that are here as well. We're going to generate things called violin plots. We're going to generate correlation plots, volcano plots, another protein abundance correlation plot, and this, the biomarker, the enrichment versus abundance plot. We're also going to go through more biological interpretation questions. After we make these plots and after we know the p-values, the fold changes and all of that, we're going to want to put in that data onto a GO-term enrichment analysis. As you might be aware, this is how we can group proteins based on their function, based on their annotation from the gene ontology website. Finally, using the things from this sheet, you can actually copy paste some of these outputs onto the website that does GO-term enrichment analysis. And finally, this part I didn't do with my code because I didn't know how to do that. But I encourage you to look into it. Okay, so everything you need if you don't want to deal with the code is in this sheet. So I encourage you to follow along. Okay, now let's get into how to do the code. Again, I've downloaded the files, this with the extension of Jupyter. This is a Jupyter Notebook extension. So I want you all to have this downloaded in your computer. And what we're going to do is I want you all to type on your browser, Google Colab. The second entry, you're going to click on it, and you're going to open. Actually, I think it's open. You're going to open that Colab. I'm going to give you time to do that. Again, that was typing on your browser, Google Colab, choosing the second entry or whatever the Google Colab entry is, and open a notebook. Okay. Next, we're going to hit upload. And you're going to drag and drop this file with this extension onto the browser. Whoops. And there is the Python file. Because how Google Colab works is that it gives you a computer, a remote computer from who knows where, that has all the specs that you need. It has Python installed. It has all the packages that Python requires for this vignette already installed. So you don't need anything in your computer except to follow along. So I'm gonna give you a couple minutes to load in the notebook. In addition to the notebook, you will also upload the raw data file, because this is what we're going to do to process. So you're going to hit this icon with the folder, and this is just your local folder and your computer, remote computer. And again, all you're going to do is drag and drop. So my raw data file is going to drag it here. It's gonna give me a warning. It doesn't know where this file comes from, but you're still gonna hit OK. And your raw data is now in your virtual computer. Before we actually get into the coding, I do want to go over our learning objectives for today. What I want you all to take away from this vignette is to understand the basics of data manipulation and visualization in Python. So this is how we extract the 2 dimensions that Simone talked about. While we do these transformations, I want you all to understand why we're doing it. Why are we doing a log2 transform? Why are we doing normalization? Why are we doing imputation? And finally, I want you to kind of get the intuition of how these different transformations impact your biological interpretation of the data. Okay, so we have uploaded the data, and one neat thing about these Google Colab notebooks is that you can run if you're familiar with Bash, you can run some of those Bash commands. So if I want to make sure that the file that I want is present in my current directory, I can type ls, which stands for list. And to run a cell, you can hit Command Enter or Control Enter, or you can hit Display button. And then it's showing me that it's listing the files in my current directory. And there you go. The data file that I wanted, raw data, is in there. So let's get started. The first thing we're going to do is we're going to import all the modules that we need. We have pandas and numpy. They take care of all things that have to deal with numerical transformations. Same thing with math and stats. Again, just math stuff. And then these 2 other packages are for plotting, which we're going to do that a lot in this vignette. So after you've imported all those files, you ran this, you're all set up to go. And the next thing is you're going to load in your data. Here, I will get an error, and you probably might, depending on the name that you give to your raw data. As you can see here, I have raw space data, and here I said look for something called raw underscore data. So if you do get the error, change that to the appropriate name, hit Command Enter, and your data is loaded. Let's take a look at this data. It should be very similar to what we see in the Excel sheet. Again, we have the accession numbers, we have the description, so this is the metadata, and we have the actual numerical values upon which we will do the transformations. Because I don't want to do math operations on a column with letters, it's gonna throw me an error. I want to remove those descriptive columns from my data set. I have delineated the name of the columns that contain the values. So here, the columns that contain the values are CTRL 1.1, 1.2, 2.1, 2.2, and HSV with the same number pattern. We have selected what those columns that contain the values are. We've also selected the columns that contain metadata, and those are the ones that have this name, description, gene symbol, and organism, right here. So we're going to separate the data. We're going to make a value data frame. That's what the DF stands for. So we're going to make this value data frame that subsets the raw data with only the columns that we care about that contain the values. And we're going to make a meta data frame with metadata that contains the metadata. So here, I've defined this function that we're going to use a lot. And I'm going to skip over it because plotting in Python has its little tricks. So I just defined this function that will let us plot via lint plots. And we're going to keep reusing this function as we do our different types of data transformation. So again, I want you to run the previous cell to define the columns and to make our value data frame, and then run this cell to define the function that plots via lint plots. We're going to run that, and that has been defined. Now, the group names control HV violent plot. Here I've just defined the names for the controls. As for the groups, as you can see here, we have this abbreviation of CTRL 1.1, and I kind of just wanted nicer names, so I just made a list that contains the names that I want to be displayed in my volcano plot. So instead of CTRL, we do control, and this is just a list of names. And right here, I'm going to show you how the raw data looks using a violin plot. So this is our violin plot, and how you read a violin plot is that the width of this little bar represents how dense it is, how many values are there around that. So we can see that there's this bulge pretty much everywhere of around 0. So a lot of these proteins have a abundance value of around 0, and we have this massive, massive tail that can go up to, actually it can go up to 1 to the 8, but the volcano, the violin plotter didn't want to go up to like 10 million, so it just stopped here at 6. But we do have some proteins that have an abundance that is humongous, just as Simone explained. We have this massive dynamic range, and how we call this is proteomics data and most other omics data, they follow what's called a normal log, positive log skew, which is exactly what you're observing here. So this type of visualization makes it harder to identify small little nudges, because if you go from, I don't know, 0.1 to 0.2, it is a full change of 2, but because it's so small, you won't be able to actually visualize it. However, we know that this data has this positive log skew, so what we can do to fix this skew is to apply log onto the raw data. So as you can see, most of the raw values are around 0. There are still some values that are much bigger than that. And this is why oh wait, I did even write it here. So if you ever forget, that's the name of it. So let's actually apply this log transformation. So what this cell is going to do is it applies the log2 function onto all the values of our value data frame. And here we're going to plot it. So when we run that cell, we see that we fixed the positive log skew. Now everything is within a similar range. And how you interpret this also is that, actually, no, that's it. So we basically fixed the distribution. Now we can do proper comparisons of the data. However, as you can see, some of these violin plots are shifted up and down. Some of them are lower, some of their little like bulge is lower than the other. And that is also going to hinder our comparisons of the data because the violin plots are shifted all around. So now what we want to do is to make it so that all the violin plots have their mean at 0. That way, it's all comparable to each other. And this is what we call normalization. So now the data is symmetrical. We will center the values for all the conditions. Values that are closer to the mean will be around 0. And we're going to apply this on every column. So I initiate a variable called normVals. This is just what's going to store a data frame or the table. And I've made it so that the indices, so the labels for the rows are the same labels as those present in the previous data frame. And the columns are the same columns as what was in the previous data frame. We've initiated this, and now we're going to iterate or do a for loop along all the columns. And for each column, we're going to calculate the mean, and we're going to subtract the values from the previous transformation, the log2 transformation, from the mean. So we've essentially centered all the distributions. And what this looks like when you run the cell is this. Now, all the means fall at the 0 value. For those of you who are following along the Excel sheet, what we've done so far is these. So we took raw data, we logged it, and this is what the Excel sheet is doing. If you can log the data, do it. If you can't, if it gives you an error, keep the cell blank. And you've done that column-wise for all the columns. So we're following along the Excel sheet. Everything's good. Same thing for the normalization. If the cell is empty, keep it empty. If it's not, subtract the value from the average of that column. So now we're here. Next step is to impute. And what imputation refers to is how do we deal with the missing values? These missing values are definitely going to affect our interpretation because we can't do pairwise comparison if a value is missing. So the underlying assumption of these imputations, as you'll see both in the Excel sheet and in the code, is that missing values we are assuming are those that fall below the detection threshold. So we make the assumption that the detection threshold is 2 standard deviations below the mean of those abundances. And then we're going to fill in these missing values with a random number that is below that detection threshold. And how that looks like in math is, so here we're going to, so in the code, again, something that you always do when you code in either Python or R is that you initiate your variable that's going to store your results. So we initiated that. And again, same indices as the previous one, same column as the previous one. And for each column, we're going to calculate its standard deviation. And this code is a little, I try to keep things short, but I might have ended up using kind of complicated functions. But what this mask function is doing is that for every value where the data is null or empty, it's going to replace that empty value with this value. And this value is, as I explained earlier, a random value that is below the detection threshold. So we've said that the detection threshold is the standard deviation, and we do some, I'm gonna say, not as relevant math for this purpose. we assume that the detection threshold is 2.5 times lower than the standard deviation. And then we grab a random number from 0 to, actually from 0.3 to 0. We just draw a random value. And then we just shift that random value down to below the detection threshold. This part is a little, I had some trouble understanding this part, so I will accept any questions from here, but this is how you do it in Python, and how you would do it in Excel is similarly, you draw a random value from a uniform distribution, you shift that random value down 0.3, and then you shift that value even more to below the standard deviation. You run this cell, and we look at the volcano plots again. And you'll notice this bulge. And this bulge is exactly what we expect. We have drawn a random value from 2 standard deviations, and we've just filled it in. So now all these random values are what used to be missing values. And we can confirm that they are all below the distribution of the protein abundances. After this, we're going to do volcano plotting. But before I do that, I want to pause a little bit to both give you time to run the cells and to ask any questions you might have. Yes. That's a Simone question.
So Ritesh asked, what is the logic of multiplying the random value by 0.3, right? So is it clear for everybody, like why we impute values like moving 2 standard deviations to the left of the distribution? Right, so we're after, as Juan said, these data are positive log skewed, right? Which means that if you take the logarithm of this data, you're looking at normally distributed data. And normally distributed data, they have a specific property that they have a width that is number of standard deviations. So 2 standard deviations, left and right, correspond to about 95% of the distribution of your data, which means that if we move 2 standard deviations to the left, there is a minus sign in front of the formula, wherein that 2.5% of the low abundance detectable proteins. That is the area that we define as background noise. So there, in Excel or in Python, we add this random number, right? And a random number, when you use the random formula, it creates a value that is between 0 and 1. But because we are working with logarithm, between 0 to 1 is a very wide distribution, right? So you are basically imputing a value which has a very large variation, right? Because it's a big chunk of the graph. So we multiply that random formula by 0.3 so that we actually narrow down the variation of that imputed value, specifically 0.3, because based on the many proteomic experiments that we do, that is more or less the intrinsic variability of a proteomic data set. So that's a very good question. But you can actually calculate your own if you want to do more accurately. Yeah. So 0.3, what does it mean? It means that when you impute a value, right, you are in that range of the graph. So that value will probably be -6, -5, in that range. If you multiply by 0.3, then you squeeze the range where that value is imputed. Now that value can only be -6, -6.3, just within that range only. Because we don't want the imputed value to be too variable, right? Because then we overestimate the variance of values that are not detectable.
Thank you. Yes. I think theoretically, because the distribution does follow a norm, because, oh, yeah. The question was, should there be values from the data that are below the detection threshold that we've set as 2.5 times the standard deviation? I'm going to give you my response, and Simone can agree or disagree, but I think that, of course, there's a chance. Of course, it's a normal distribution. It could be, but we made it so that it is unlikely that there are going to be values below that because we've looked at the standard deviation. We've made it so that there shouldn't be that many values below that. Is that right?
Just, yeah, I completely agree. Just as a tiny additional note, when we impute values, we're always making up numbers, right? So whatever number you put there is wrong, right? So now the question is, what is a wrong number that represents more or less the enrichment that you want to calculate? The reason because that little bubble of imputed value is there and not massively at the very, very low end is that if you create as imputed values that are really, really tiny, and what happens is that when you have proteins that, for example, they are barely detectable in one condition and not detectable in the other, if the non-detectable values they are imputed, you get a fold change that is massive, right? Because you have detectable versus non-detectable. And that actually tricks you when you interpret your data, as it looks like proteins that are really enriched where they are barely detectable. You don't want to encounter in that risk. That's why, obviously, wherever you put an imputed value is always wrong, right? Because it's imputed. But the reason because we put them there is because it's a reasonable statistical estimation of what is the background noise of our data. Yeah. Yeah. Yeah, so the question is how many valid values is good enough, right, in order to estimate the fold change. So interestingly, and again, this is arbitrary, but interesting, we actually don't set a threshold. Like, so even if we have one replicate quantified out of 4, we still keep that protein with us and we calculate the fold change. The reason because we're not too worried about it is because if you have a protein with 2 valid values and 2 imputed one, or 1 valid values and 3 imputed one, the variance of this measurement is going to be so large, it's never going to be statistically significant when you compare the 2 conditions. So yeah, you will have a fold change, but it's going to be very low in the volcano plot, and it's not going to be a protein that affects your biological interpretation. Yeah, basically that protein, regardless whether you put your threshold, will be towards the butt of the volcano plot, like in the very low part. And so, yeah, whether that protein has valid values or whether that protein you throw it out, in the end, in your biological interpretation, is not going to make much of a difference.
Oh, good. Thank you. Thank you so much, Simone. Okay. So now that we have processed our data and made it all nice and good, we're actually going to be doing some interpretation. And this is where the coding might get a little bit more intense. So again, encourage you all to follow the Excel sheet. But let's generate a volcano plot. And there are a couple of considerations we're making a volcano plot. So we've transformed the data. There are no missing values. We will identify those proteins that are significantly differentially expressed from control to the infected cells. So first, we're going to calculate the average protein abundance within each condition. Then we're going to calculate the fold change of the average HSV expression compared to the control group. Usually we think of fold changes as one value over the other, but because we have already transformed the data into with log using our log identities, this translates to log of one value minus the log of the other value. So we're actually going to be subtracting average expressions to calculate the fold change. Next, we're going to determine which proteins have statistically similar variances, and these are called homoscedastic. These are homoscedastic proteins, so the ones with similar variance. And we're going to also find which proteins are heteroscedastic or have unequal variances. The reason for why this is important is because we are going, well, I'm getting a little bit ahead of myself, but to do this, we're going to do an F test. And the reason for why this is important is because when we calculate our p-value, we're going to use an independent t-test. And one of the parameters for a t-test is to tell it, are these two groups with similar variance or without similar variance? So that information of heteroscedastic and homoscedastic is going to go onto our calculations of the p-value. So we're going to, and yeah, this is where I explained that. And then from there, we're going to calculate the score, the protein score. And this is just the product of the fold change times the log 2 of the t-test value. Though I think it's actually the log 10. So let me log 10. Okay. So the log 10 of the t-test p-value. So that's how it looks like in Word. So let's do it in code. And so first we separate our data into control and HSV, and we do this by knowing the name of the columns. So we've grouped the name of the columns based on their names, and we put them into two different lists, called control cols and exp cols. We're going to, as always, initiate an empty data frame. We know that it has to have the indices the same as the previous data frame. And we also know what we want the columns to be: the average control, HSV average, fold change, the F test, negative p-value, and the score. We've initiated that. Then we're going to start filling in those columns, column by column. So first line, easy enough. It's just the average. So we just compute the mean along the first axis. So we just compute the mean for every row. So, and that is going to go onto this column. We do the same for the HSV averages, calculate the means, and we're going to calculate the fold changes, which again, easy enough. We just subtract all the values from this column from the values in this column. Next, we're going to determine if the variance of protein values are the same between control and HSV infected groups. So we have separated our value. These are the variances of each group. So we've calculated the variance for the control, the variance for the experimental, and we calculate something that's called the F statistic, which is just the variance of one over the variance of the other. And now we're going to actually do the F test. This is, again, a little bit more involved line of code where I'm using what's called vectorization. I've taken a function and have told Python, turn this function in a format so that I can apply it along a column. If you have more questions about this line of code, if you are proficient in Python and want to learn more about it, feel free to ask me. But the gist of it is we're just calculating the F statistic for all pairs. Oh, sorry, we're calculating, the F statistic for all the F stat values, okay? And we're gonna store that into this column called F test. So these are p-values. Now, using that F test, we're going to conduct the standard t-test for proteins with the same variance. And the name of the t-test for things that have different variants is actually called the Walsh's t-test. And this is why it's important to differentiate the two. And so now what we're doing is we're looping through every index. So these are all the proteins. So for each protein, we're going to determine if the F-test is lower than 0.05, and this tells us if the variances are unequal. I think the null hypothesis, I don't remember what the null hypothesis is, but yeah. And we're just going to, if something is below that p-value, I think that means that the variances are unequal. I might need to go back on that. Okay, thank you. And then we're going to conduct an independent t-test using that information. So equal var becomes a Boolean, either true or false. And then we're just gonna pass that variable onto the function. So the t-test function asks for the control values. It also asks for the experimental values. And it also asks for, are the variances equal? And we've just provided all of that. And the p-value from this test is going to be stored in this column. We have the p-value, we are going to transform it onto the log 10, and we're gonna turn it into a negative value just because these p-values are small. So when you do the log, they're gonna be negative, and we want them to be positive, so we just negate it. So now we have a bunch of columns up to here, the negative p-value. And all that's left to do is to calculate the protein score, which is just the product of the fold change times the negative p-value. What this looks like on Excel is identical. We have our average of each expression. We have our fold change. We have our F-test. And I think I actually don't, yeah, but this does what we want it to do, and you can follow along as well. We've calculated the score, and now what I am showing you here is the distribution of these scores, just so that you can get a sense of what these scores look like. So let's run this cell. It's doing all the calculations. We can see that most of the scores are around 0. And so, and there are a couple, there is a little bit of a tail right around here. And this is just to give you a sense of how do these scores look like. I'm gonna take another pause here and be open for any questions. Can you please read them to me, or actually read them on the mic?
One question is why generate, I guess, data from uniform distribution while assuming a log normal? I'm just reading verbatim. Why generate from uniform distribution while assuming a log normal?
So the question is, if we are, how are we assuming that data are normally distributed?
Values, we assume the imputed values come from a uniform distribution, but the entirety of the rest of the data follows a normal distribution. So why the disparity?
Yeah, so I think if I understand the question correctly, yes. So if you look at the distribution of the protein values after they are log transformed, these are normally distributed because they look like a Gaussian. Whatever test you do is going to tell you these data are normally distributed. However, I think the question comes from the fact that we are using the t-test, which starts from the assumption that data is normally distributed. So here's the thing. In theory, the t-test is not the ideal test to use when you compare data of which distribution is not known, right? You should use a non-parametric test. In practice, non-parametric tests, they are based on rankings, right? So the reason because they don't care about what distribution you have is because the values are substituted with ranks, and then you compare ranks of group 1 with ranks of group 2. When you have 3 replicates or 4 replicates, if you're using non-parametric test, you lose all the power in the statistical analysis. If you think about it, if you have only 3 replicates, you have only a very limited combination of ranking. Like you have 1, 2, 3, 4, 5, 6, or 1, 2, 5, 3, 5, 6. Like so, it becomes like very limited, right? So if you use a non-parametric test, the equivalent is like Mount Whitney, for example, it's not going to work with 3 replicates. So we do a t-test assuming that our 3 replicates or 4 replicates have a normal distribution. We cannot calculate that. The formula to use it, the test to use to calculate the normal, whether distribution is normal, is the Shapiro-Wilk. There's probably a formula in Python that you can use. It doesn't really work when you have so few data points. So yes, using the t-test is an approximation, but it's the only one who has enough power to deal with so few data points. I think that's probably the question. It was the only question? Oh.
All right. The other question was, is the reason you're choosing negative 0.3 because it's the widest point in the violin plot?
No, actually, honestly, like we discussed before, wherever you put your imputed values is an arbitrary choice. You can go 3 standard deviations to the left. You can multiply by 0.3 or minus 0.3. Honestly, it's your choice. Nobody's going to tell you this is wrong, this is right. What you have to be really careful of is to not trick yourself, like so to not impute values that are so tiny that your fault changes will become overestimated or so large that you're not going to be able to tell differences between existing and non-existing values. The numbers that we put there is what they usually work best for us. If the shape of your volcano plot looks a little bit too weird, this is something you can work with. Yeah.
One more question in Zoom. So could you use the result of the t-test, for example, the t-score, instead of computing a score based on the fold change?
The score, did you actually talk about the score? I don't remember.
Yeah, just now although the score is going to come back when we make the plots. So
Yeah.
maybe the question will be more relevant and probably explained after we finish the vignette.
Yeah. Juan is gonna explain you well what we intend by score. And again, also this one is arbitrary, but it's very practical the way we design it. So I'll let you go ahead.
Yeah, so going back to the score, like again, it is arbitrary, but it is a helpful metric for what we'll be doing downstream. Also, I don't know if, I think we did announce that I have to go at in a couple minutes before 1:30. So I will unfortunately have to speed up the last couple parts, but all the code is here. So I do encourage you all to play with it and see what types of different interpretations come from different ways that you analyze the data. But now we have extracted a bunch of very relevant information from the data. The fold change, very important. The score, very important. And the negative p-values, which again, are very important. So now we're going to visualize all these features we've extracted from the data. And so the first thing we're going to do is to identify or compute the correlation between different values to assess the likelihood of there being batch effects. As we talked at the beginning of the slide of the vignette, there are these experiments were conducted on different days. So we do want to see the influence of that. And to do that, Pandas, the data frame, has this neat little function where you can just plot or calculate the correlation between the means of each column, and you can easily visualize that with a heat map. So right here, we have calculated the correlation between all the values in HSV2 and seeing how they correlate with HSV 2.1, 1.2, so on and so forth. And again, the diagonal does have the highest correlation because they are identical. And then we do have similar clustering among the things that have the same condition. So control values have higher correlation with other control value. Control value. Oh yeah, lighter means higher. Control values have higher correlation with other control values, lower correlation with the things that are infected, which is what we expect. However, we do see higher, I think, well, with the scale of the color, you actually can't see it. But if you play around with the scale of this, you can see that there is indeed a batch effect. For example, here, these two things, control 2.1 and control 2.2, clustered together. These are hierarchical clustering that is being done in the background of this plotting. And we do see that there is some clustering with things that come from the same batch. So indeed, there is a batch effect, and we can nicely visualize it with this plot. Another good plot to calculate is this volcano plot. To do this, again, plotting in Python does require a little bit of a learning curve. So I just defined this function that will make your volcano plot and will give you nice colors for it. And here we see our volcano plot where we have drawn two horizontal lines around the fold change. So this is the horizontal line that's this is the vertical line, this is the horizontal line. We've drawn two vertical lines around the negative 0.5 and positive 0.5 fold change. So these are the ones that have a log2 fold change higher or lower than 0.5. And then we've also drawn a horizontal line for a cutoff for the p-value. And this is from a Bonferroni correction, as you may be aware, because we have so many different tests we want to correct, and we're just doing it with a simple Bonferroni correction where we divide the 0.05 p-value by the number of proteins, the number of tests we have. So we've drawn a horizontal line, we've drawn, horizontal line, two vertical lines to delineate the proteins of interest. So as Simone talked about earlier, we see that there are a lot of things that are significant, and these are the ones that are reproducible. And the proteins here have do have a high fold change, the ones in blue, but they are not reproducible. They don't have a low enough p-value. Another neat trick that I used for this plotting is that I have marked which proteins come from human, which proteins come from herpesvirus 2, and which ones come from herpesvirus 1. And the legend is here, and we can see that a lot of the herpesvirus proteins are enriched in the herpesvirus infected cells because, of course, herpesvirus is there. And we can clearly see that here. Another important useful plot is calculating the protein correlations. So this is how each dot here is a single protein. And where that protein falls is the average expression of that protein in the control and versus its average expression in the infected cell line. And I've also color-coded the dots to represent the score, as you remember. Again, things with higher scores mean that they have both or either higher fold change and or lower p-values. And so, again, we see that all these proteins that have very high scores, so we call them like the things that you want to look at, the more relevant proteins, are have almost no abundance in the control, but have high abundance in the HSV infected cell line. And we can clearly see how things with also lower scores, which are also things with either or high abundance and high or low p-scores, p-values also fall around here. And the things that have around 0 score. The ones that are not as statistically significant are light values, and they do tend to fall closer to the x equals y line. Another important plot that Simone mentioned earlier was the biomarkers, where we want to observe if things or proteins that are enriched in HSV, if they have also higher log fold changes. Again, each dot represents a protein, and where they fall represents their fold change and their abundance in the sick condition. Finally, I am going to, this is the last section, and then this is the part that you can do on your own. And this is where we are going to rank the proteins based on their score. Things with the highest score are the things that you want to look at the most, the more relevant. But we don't want to throw away things that are below some arbitrary threshold. We want to look at how are things with higher scores influencing the functional landscape of your cells. So we have ranked our proteins based on their score, and then this is our list. And all this code is doing is that it's going to export this ranked list into a text file. We run it, and I don't know where it went. Oh, you think so? Oh, there it is. So in our folder, and then this is our text file with all our ranked proteins. We select all. Now we're going to go to this link, Gorilla, and this is what is going to do our GoTerm enrichment analysis. Click. Okay, and then Homo sapiens, blah, blah, blah. We paste. Okay, Control C. We pasted it onto this server, and we're gonna go and run GoTerm enrichment analysis, which choose file, search enrichment GoTerms. Does a bunch of math in the background. And now we have a map of the enriched and relevant GoTerms from our data, from our ranked data set. And here's where I encourage you all to look at this map and identify the different functions that are the more, I would say, statistically relevant, statistically different from control to infected. And we see a lot of these functions are enriched. So the ones that are more red are the ones that have a lower FDR score. So the ones that you should probably look at more closely. And we see a lot of reds. So we have mRNA metabolic process. We have more nucleic acid, nucleotide metabolism functions. We have, again, more nucleotide metabolism. And you can just go around, and this is what is going to guide your interpretation of the data. This is a good first pass of what is going on, what is statistically different, and where should your story start. This data would tell us that there is, you should be looking closely to how these nucleotides are being metabolized. And this is just how you can start and keep exploring your data. I think I unfortunately have to cut it short. I have a class that started 2 minutes ago, so I'm going to excuse myself, but I do encourage you all to look closely, play around with this. There are different ways you can rank this list. You can rank it based on the abundance of only the control, rank it on the abundance on the HSV, and look at how these different proteins, the function of these different proteins. With that said, I'm going to hand it to Simone, who is going to answer some questions, I think, maybe.
That's great work, Juan. Thank you so much for, he put together all the scripts. Like, I don't know how to code, so it's all his work. Yeah, but I mean, I think we can, finish here because I don't want to, overuse your stamina. I don't know if there's any final question, but as you will see from the Excel file, it's divided in sheets exactly because the first part are the data processing and transformation, what you guys have been hearing for the last hour. I think in the last sheet, if I'm not mistaken, we left you a few like biological questions. And based on what you heard today and based on the formulas that now you have access to, you actually should be able to answer that. Like you should be able to use the tools that we provide you to tell what is going on in these cells that are being infected, what kind of pathways they activate. Is there something that we should know regarding how much protein mass there is in terms of viral content versus like human cellular content? I actually forgot all the questions that we put there, but to sum it up, it has been probably tedious, but you guys now, you should be able to take a raw proteomic table and end up with interpretation. Right? And obviously there are many more graphs and figures that can be done, but we thought about this workflow exactly because this is what in 1 hour can be done. Any final questions? No? That's perfect. Yeah, so thank you for coming. Is this the last Omics Club lecture? So I let Alex make the conclusive remarks.
So thank you very much again. Let's give one more round of applause. And yeah, really want to thank you all for your attendance and for your continued interest over these last 7 weeks. So it's a really, really amazing. And please give a round of applause also to the Omics Club board, who spent a lot of time in meetings and sent so many emails. And yeah, we really appreciate your time. And yeah, could the Omics Club board people please come to the front? Let's take a quick picture and then we'll call it a day. Thank you. A question? This workshop series is the first time we did this. Yeah. We'll see. I think it's something that maybe the school will try to institutionalize and probably do better than what we did. But right now, you can check the recordings.